All right. Yeah, so this is actually not only from myself, but also from some people of my lab and Sebastian as well, who's here on the call. So actually, I want to give two studies or two things that pertain to what we're discussing from the front line, so to speak. So how that works out from our perspective as working scientists. So I have to give you a little bit of background of what we actually do. So you've all heard about genomics, or you know about the genome that we have, and then that gets translated into a transcriptome. And so I'm of course writing it, presenting this way around. So the genome is a little thing, but that's only there to actually instruct all the proteins that are being made, and the proteins are the ones that do something in your body, and they can also be biomarkers, which will be the topic here. So this is another view of biology. So this is the cell, and the cell is, so it's like a cross-section of a cell, and this is actually where the genome is in the nucleus. And but everything that happens in the cell is actually done by proteins and their dynamics, which is sort of illustrated here in cartoon form. And that's not really important, but for the discussion, but that's what we analyze, and we develop a lot of technology and generate a lot of data. And then we've been spending the last many years or even decades on more cell biology problems, and now we want to use it for clinical applications. And then that is where we have to face a lot of these issues that are the topic of our workshop. So this is to give you a picture. So this will be a particular structure in a cell with cryo-EM and microscopy. And what we bring to the table is then we could say what are the proteins that make up this structure, because all these structures are actually made up by proteins. So then we have, similar to sequencing facilities, we have these high-tech facilities where we have mass spectrometers, and they record the proteins. So we have a complex workflow where we can then, our goal is to record as many proteins as we can in the system and learn something of that biological system. And then nowadays also about some diagnostic applications or diagnostic use. So first part of my talk, I want to say how this came also up this morning. There's a big shift in how you actually develop software, and which is now shifting to a much more collaborative and less proprietary mode of doing that. So so we develop a lot of software. Most of science is also working in a more in silico direction. So software is becoming ever more important, and scientists have actually pioneered a lot of the things that then led to the world we now live in. For instance, and not least, the internet. So that was actually developed at CERN, or some part of it at least was developed at CERN, as John Norton has written about. And I think also the open source is another area which could have huge implications even beyond software. So so open source was actually the way things started initially, and people were very angry at Microsoft in the 70s that they actually charged money for software. But then of course that flipped completely, and as we heard from John, Steve Ballmer of Microsoft likened open source to a cancer. But actually that has changed dramatically even for Microsoft. So the big event was that Google had this TensorFlow library that enables you to do the deep learning, so the artificial, or the last version of the artificial intelligence, and they actually open sourced it. And that was very unexpected, and this is like 8 years ago or something. And why did they do that? So they thought, drawing on the community would be actually good for them and for the community. So I do think that, this is not, don't be evil. is actually, you know, companies can be at least more or less evil, and Google is one of the less evil in my view. So anyway, also, so this was partly altruistic also, and then we can use this library, or anybody can use it to build on top of it, right, and to improve it. And the same, Facebook made an even better library. It's called TensorFlow, and Microsoft again has made an infrastructure which we use to develop code in, called VS Code. So all these big companies are doing that, but of course the scientists themselves, and also in other fields, amateurs also do this, and there's a whole infrastructure on the right-hand side, GitHub, where you then deposit your work, and this has a lot of intellectual property implications, and they are good for us because it's generally, if you put it on GitHub and make it available, you're much less likely to be sued, even though you could argue that your source code violates certain patents, but this almost never happens. And people have, they're making all these tools that make the whole process much easier. And it also goes in the direction of open science, reproducible science, and that you can build up on things that others do. So seems to be stuck right now. Okay, so and then this is also something very good. So this is a particular person, so it's or even a couple. That so Jeremy is and Rachel. They are two of my heroes. So they are at State University of San Francisco, and they are just very few people, and they have also in the open source spirit. They've developed this course and this software library called Fast AI, and their slogan is making neural nets uncool again. Because as we heard, there's a big apprehension that Google and Facebook and also the Chinese companies, they will develop these advanced AIs, and we don't understand them, we cannot use them, and we are just sort of subject to them. But they are showing very effectively that you can actually build on top of these open source libraries, and then you can actually, without a PhD, you can actually have state-of-the-art neural nets, and so this whole drive to make to democratize AI, and I think it is very successful. Also, Jeremy is actually, that's in his data scientist capacity. He was one of the first ones in the US that picked up on what was already mentioned, that the Asian countries that have masks, they actually had much lower COVID infection rates, and then he got really into this, and he's one of the individuals that really tipped the debate in the US early on, which actually didn't happen in Denmark. So Denmark, they still run around without masks. And then also, this is Rachel Thomas here. So she has a course on data ethics, and it's very good. I encourage you to look into this, where of course you hear all these terrible things that can be done, and have been done with artificial intelligence and the social media, but also maybe what we can do about it, and just exposing it, right? So these, there is at least some individual efforts that are highly effective. So coming back to what we do in the lab, so this is another colorful view of the tools that are out there. So we use this Python ecosystem. It actually comes from a programming language that was meant for children. But then it developed into this due to open source and all unpaid actually. They made this tools, like this was just published after 20 years in Nature, and we all use them all the time. And it's so the shift has been away from commercial solutions to open source, and they're all free, and we use them all the time. So we can use it to build on them. So we work much faster then, and also due to the fact that a huge community is using them, we're much less likely to make mistakes than otherwise. So and then so and then why would we do this? So we want to, of course, then give back to the community, but we also, when we expose our code base, then many people will look at it, and they may test it, and and improve it and point out errors. And it makes it also quite, there's a whole system that everybody can actually contribute. So then it has to pass a quality test, but actually everybody can contribute with so-called pull requests. And then it's like, okay, errors will be made, but then we can quickly correct them. And also there's documentation. You can follow everything back, and we can document it, and everybody can build on top of it. And then as a scientist, you'd actually also share the code and everything that increasingly affects society, like the code that the big tech companies use. You could argue they should actually be open source, because otherwise we don't know what basis these decisions are made on. This goes not only for the big companies, but especially also for governments. So again, so we use this, and we have a particular project by Maximilian Strauss here, or led by him, called AlphaPept. And then we use this infrastructure. So the details are not important, but we can build on this whole infrastructure, just write the code we have, and then it goes in, and we can develop the software with the same rigor that big companies will actually develop it in. So yeah, and so this is our code base here, and then this automatically gets tested, and this allows for people in China or in Africa, wherever, they could also actually contribute. So this sort of goes in the direction of citizen science, and the other way around. This is an example of this Picasso software that Max wrote. Then sometime later this year, tool came out, and he could just build on top of that and incorporate it, and 3 months later his software was much better. So this shows, so this is then the way this works. So this is for this Picasso software. So you can see who contributed here. So the higher the peak is, the more contributions at that point, and you can see for everybody what they contributed. So this is a very interesting model. So it's a sort of a star-based, so people even compete, you know, how much they can contribute to these things. And there's only in this project, this small project, 5 direct contributors. But if you then look on what that depends on, so you can argue there's 22,000 people whose work they somehow aggregate here. So and then yeah, and then this is actually now beyond software. So this is also by crowdfunding being extended to hardware. So there's no reason this actually has to be only for software. It could maybe also apply for government things. I'm not the expert. Okay, so that was the first part. Second part is maybe something more close to home. So we, or this is just how we develop the knowledge mining. So apart from this other software, we also do something similar to Google. I mean, Google also didn't invent this, but we do huge networks, and we aggregate all the information so that we can better say what put what we found in context. So this is what they also use, the big tech companies, but we use it for medical gain or learning more about biology. So what you see is on the left-hand side is what the data we generate here, and then on the right-hand side we put this in context with literally millions and millions of data points that we can download for free from the internet. I mean, these are usually government or intergovernment sponsored efforts that have led to this knowledge bases. So, and then we hope that this can be used, or we're already doing this in sporadic cases to, for instance, make a better cancer diagnosis. And that's where we first got into these ethical aspects. For instance, when we have late-stage cancer, right, then we have the issue that people are desperate, so they will try anything. So if we say, here's a new target, proteomics has found a new target, they will just immediately go for it, right? But that's maybe not ethical because the chances may still be very low. Okay, but the main thing where this now comes at us in full force is with something called plasma proteomics. So this is sort of a cross section of your body with the blood system. So blood system touches every organ. And then if there's a damage in one organ, this can be reflected in the bloodstream, right? So let's say there's some heart proteins, and when the heart is damaged, they will be released to the circulation. And if we analyze the proteins in the blood, we could say you are at risk for heart disease, right? And this is from the clinic here in Munich. So there's 9 million tests actually, and they are mainly done actually on proteins or on small molecules that also float around in the blood. So our goal is to measure, and that's where also the ethical issues come in. So normally these are one protein at a time, right? Like I said about the heart protein, so they would look for one heart protein and tell you you've had heart attack. But it's in the nature of our technology that can measure many proteins all at once. And then we want to have a generic test. So we not only say, okay, you have maybe risk of liver disease, but also of heart disease, or maybe even of a psychiatric disease. So, and this is, yeah, this is also not, we are not the first people to think of this of biomarkers, but this is an infamous example of Susan Holmes that had a big company that also wanted to diagnose people on one droplet of blood, but then it turned out to be all fraud. But anyway, she didn't use mass spectrometry, so we use mass spectrometry. This is headed by Philip here, spearheaded. So we take a drop of blood, and then we get a measure of many proteins in the blood, and then we want to use this. So we have done this now in many studies already. So for instance, alcoholic liver disease. So we can follow people with alcoholic liver disease, or that may be at risk of alcoholic liver disease. So we can maybe tell them early. And also when people have had bariatric surgery, this works for some and not for others. And in this case, it's not blood, but for Alzheimer's disease, we want to find a biomarker of the state of the Alzheimer's disease. And here's a newborn study. So we are studying, in that case, the whole blood of term and preterm babies. And if that looks good, they could be sent home earlier. And if it doesn't look good, they have to stay in the unit for longer, right? So these are very practical things. And if they worked as advertised, it will be really, really good, because, for instance, in this case, if they're released too early, they may have a handicap in brain development for their entire life. So this is this newborn study with many participants. So we get a lot of data. So it's in the nature of this thing that we get a lot of data. That's what it thrives on. And we can, for instance, look what correlates to your body mass index. You know, this symbolizes the levels of proteins in your blood that correlate with your body mass index, right? And this is just the way we show it and what proteins these are. And for instance, you can see the C-reactive protein here. That's the marker of heart disease risk. And so what this is telling us, that with a high body mass index, you tend to have high levels of this protein that indicates a bad status for your heart. But then this goes for all kinds of things. So we have a lot of meta information. So this is also another study that we're about to publish, where we actually smack ran into this ethical implications that I talk about presently, because we have done it now, everything's finished, but we're not allowed to publish it. So this is the case for liver disease. So we are, so 30% of the population has steatosis, fatty liver, caused by obesity or alcohol or both. And then this in a subset of people that goes to fibrosis, cirrhosis, and death. So we want to be able to say at an early stage, are you on a bad trajectory or not? So we've done this, and this shows machine learning. So take home from this is if we can now do this better than the best liver test that's out there. So so far so good. So our vision is that we'd actually have a generic test, and so we would get a lot of data of the nature I just showed you, and then you just give us a drop of blood, we analyze it, and we can say, you know, you have liver disease, you're risk of heart disease, or maybe the treatment you're taking is not working for you, and I'm a vegetarian, so maybe that's good for me, but for you it would be better to eat McDonald's or keto diet, whatever. And So that's our vision, right? So if it worked, it would have a huge impact, but it has definitely it's definitely more complicated than we thought. So when we did this, we could see, members of the lab. We can actually distinguish males and females. So whoever's above the line is male, male, and below the line is female. And we can also say whether they are pregnant and whether they are, if they're a woman, are they postmenopausal or not. And we can say, what is the inflammation status of them, which would be a heart disease risk, and apolipoproteins, that's sort of equivalent to the cholesterol test, but much more detailed. And we can also, to some degree, say there's an allele variation. So if you have the wrong allele, you have a many-fold higher risk of Alzheimer's disease, and you may actually not want to know that. Yeah, so we have done all these studies, and they have driven home to us that we actually also get a lot of information that is ethically sensitive. So for instance, just pick out this one here. So we can see whether sugar is attached to the proteins, and if it is, then that means you have uncontrolled sugar levels, and that means you are pre-diabetic or diabetic. And a large proportion of diabetics people, or people with diabetes, they actually don't know they have it. And you can eminently do something about it. Yeah, and we can read out more than 100 already approved biomarkers all at once. So this is this unexpected information that I just showed you, and that then led us actually also as a result of our work group. It led then to a collaboration. What are these? What are actually these? How should we tackle these things? Because not the one thing is good and the other one is bad, but we have to somehow find a way to preserve the good things and as much as possible, while being ethical and staying in compliance with regulations. And that is more problematic than I thought at the time. So yeah, a lot of this is from Philip and Peter, that's on the call here also, and Sebastian, who can jump in. So first thing is, look, it's actually the information detailed enough that you can identify a person with that, right? In genomics, that's the case, but for proteins, you don't think about that. But then we, Sebastian looked into this. So this is GDPR rules that you are well familiar with. So we try to see, how does that actually apply to our case? And this is actually papers we handed 2 papers now that I'm talking about that we submitted 2.5 weeks ago. And the first one deals with these, whether actually the plasma proteome is detailed enough to identify a person, and what are we finding that that we haven't even looked for. So we go through this here, and the answer is that so from the levels of, even when you're not sick, but just from in one study, from the levels of the proteins, when we then compare the samples to each other, so these are more than one sample from the same person, we only make one mistake by saying, this sample belongs to this person. So that's this is in a small cohort, so it wouldn't hold up in court by any means, but it's getting in the direction of identifiable. And we also get some of the genomics markers that are reflected in the protein sequence. And then we make also some more mistakes, but it's also going in the direction that yes, this is soon going to become identifiable. So then if we put the data out on the net, and somebody else has the genome of that same person, they have a good chance to match the two together. So yeah, and so this is what I already said, but we investigated again, so we can tell men from women in most cases. Yeah, and we can see some of these alleles that give you, they can be actionable or non-actionable, for instance, for risk for neurodegeneration. So that was in a nutshell the first one. And we also go into what would have to be the case to not make it so. So then we have to strip certain information out. but then we cripple our data set, right? So that's the problem. And then in the second one, we go into, what should we now do, and what are the philosophical bases, or what would philosophers say to that, and what would the literature also say in the case of genomics or imaging data, where they have had these issues for quite a bit longer. And this is what we can discuss if you're interested. So so what Sebastian and Peter did is they did a very good and exhaustive literature review. So where has this already come up in the context of proteomics? And they identified these issues that in the papers that people had mentioned, and so here they are. So this is a condensed version of how the issues that come in our field, how they are mapped to these issues that we would talk about in a normative context and regulatory context. And then we go through, and we can discuss this more. How do we deal with them? And maybe the first impulse would be, okay, you cannot have this, and you need to have very strict, like you should not report if somebody came from a liver disease, you should not tell them that they have diabetes, right? But that also raises issues, because now they could die or get blind from diabetes, right? So you didn't do the right thing. So it's not clear what you should do. And as a starting point, and that's similar to also what's done in genomics, we came up with this questionnaire that the person can click, what information. So they shouldn't be told probably non-actionable data in any case, but they could click which of the actionable data do they want to hear. So that's where we are, and we've sent in these 2 papers, and I want to acknowledge the co-authors, and also Sebastian acknowledges his mentors here, and some of the people we work with. So that's what I wanted to say. I hope this gives you a feeling that this is not just dry things you are discussing academically, but it is actually coming. And as I said, one of our papers is now being held up for GDPR reasons. It's the first time this ever happened. Okay, then that was what I want to say.